Good evening. Welcome to the second session of the two-day international webinar on recent trends in biosciences and biotechnology. I extend a warm welcome to all the participants for this second session. I take it a great privilege to welcome the resource person of the session, Dr. Muhammad Sahib bin Mustafa, Dean, Faculty of Applied Sciences and Technology, Universiti Tun Hussein, Malaysia. Dr. Mustafa is one of the eminent persons in the field of biosensor. He has done a remarkable research in biosensors and is now among us to share his knowledge on biosensors. Welcome, sir. Welcome to the session. We are looking forward to your wonderful, knowledgeable talk. Welcome.
Yes. Good evening to all. Today, the first day, second session. The second session, resource person, Professor Muhammad Kamarulsaki bin Mustafa, University of Uttam, Malaysia. He is a Pro Vice Chancellor of Uttam University and former Dean of the Science and Technology Department. His experience more than 18 years, and he committed the ad hoc posting 20, editor more than 5 journal, keynote speaker more than 12 numbers, project completed 8. He got the 15 crores of Malaysian ringgits for this project. Awards, he got the international awards 12 numbers. He guided the PhD students more than 8 ongoing. He reviewer of the journals, international journals, more than 25 international journals, most of the Science Direct as well as the Scopus Index. His publication more than 100 international. His citations more than 3,000 numbers.
Thank you to Madam Chairman and the organizer of this conference for inviting me to give a presentation, to give a talk this evening. So I also would like to thank to all the participants because you know, until still waiting for the last, might be the last talk for today conference. And actually in Malaysia now is around 6:30 in the afternoon. All right. So never mind. I'm looking forward to give a talk with all of you. Okay. Well, actually, what I'm going to share today is about the biosensing technology. Well, we are actually now facing about COVID-19. So I think this topic is quite interesting, and maybe we can share and we can collaborate later, which might good for our country. And well, at the moment I'm the Faculty of Applied Sciences and Technology from Malaysia. So the first, I think, keynote speaker also my colleague from actually a different department. My background is physics, so but I'm focusing on biosensor. Well, when we talk about, I try to give some insight about biosensors, since might be the participants in this conference come from various background. So I will go a little bit on just a very basic introduction about biosensors. Actually, in our body, there are a lot of sensors. Sensors is very important to sense, to move, to make a decision for a human. In our body, you know, for example, our tongue, there is a very good biosensors where every part of our tongue can feel different taste. If you look at you know, the difference, the area in our tongue, so there are area where we can feel the sweet, sour, salty, bitter. So this is all kind of biosensors in human body. And for example, as our skin as well, so our skin can feel the heat, our skin can feel, the different temperature. That's actually the biosensors that available in our body for the human being to make a decision, to make a move. So when we talk about sensors, especially in, we are in the IR Industrial Revolution 4.0, sensors is very, very important. Everything needs sensors to move. All the manufacturing, all the industries where they are using mechanical parts, or they are using robotic, they need sensors to make a decision. So just in this case, we are combining biology, biological, biochemicals, and sensors, so they become biosensors. So biochemical reactions is a basics of signal detector, right? So from the biochemical reactions, so they give a signal where we can detect either in optical forms, either in the electrical forms or thermal forms, for the estimation of the concentration of the analyte in the given test sample. As for example, as I said just now, if we measure temperature, if we measure temperature, the sensor or our skin, for example, they can estimate what is the temperature. Is it 24 degrees? Is it 100 degrees? So, so that is basically what the sensor is all about. But biochemicals, they give the response towards the output of the sensors. So among the various biosensors method, electrochemical presently hold a leading position. So electrochemicals, biosensors, currently there are a lot of research going on that. And in fact, the one that available in the market mostly is electrochemicals. So this is one of the reasons, is because the electrochemical sensors come with screen printed electrode. A screen printed electrode, where this one we can actually use it as a disposable, and anybody can use the sensors independently. I mean, they can use by their own very easily. No need to have experienced personnel, no need to ask for a doctor to use the biosensors, electrochemical biosensors. And it provides a very mobile, cheap and disposable biosensors. So once we use it, you can throw it away. So I think most of us experience this kind of sensors. For example, if we want to check our blood sugar content, we use we go to the pharmacy or we go to the clinics, and we put our blood on the very small screen printed electrode, and then they connect it to the reader. So that is what is electrochemical biosensors work. Okay. Next, how to go to next? Okay, next. Okay, so this is the screen printed electrode. If any one of you are not clear of it, we call it screen printed electrode. So there is a screen printed electrode is printed on the screen. So we are we deposit all the biochemicals on the working electrode, and then the electrode will the chemical will react and then give the signals to the output. So how I'm going to delete this line. Oops. Okay, never mind. So this is a zoom in of the screen printed electrode, where actually we have the counter electrode, we have the reference electrode, we have the working electrode, and all this biochemical interaction happen at the working electrode. This is actually the miniaturization of previously we conduct the electrolysis experiment where we have 2 electrode or 3 electrode inside, and then the electrolysis happen. The changes of ion happen between the electrode. This is actually what happened on the screen printed electrode. In this example, for the screen printed electrode, we can deposit all the chemicals on top of the working electrode, and all the recipes is here. For example, they put a glutaraldehyde to ensure that there is a covalent binding, and then here is about the immobilization of antibody, and then the introduction of ultratoxin detection. And then this actually the printed electrode, screen printed electrode, can be connected to the reader for the measurement. So we can put our sample on the working electrode, and then just straight away go to the reader. We can get the reading straight away. So that's how it's work in the pharmacy or in the clinic. Normally, when we want to test the uric acid, we want to test sugar level, so we can do it easily. Okay, but in order to develop good biosensors, we need to have high specificity. High specificity means that the sensors only specifically detect the disease or the one that we want to detect. For example, if we want to detect malaria, so the sensors only react with malaria sample or malaria antigen or malaria cell or malaria protein. So this must be high specificity. It should not react with other protein or other sample which is not for malaria. And second one is the portability. It must be portable because if you want to use at home or anywhere, anytime, it must be portable. So if the sensors is not portable enough, means that the patient need to go to the specific place to check the measurement, which is become the limitation of the sensors. And then the simplicity. So simplicity means the biosensor didn't require for the experienced people or didn't require for expert to do the measurement. They can do by themselves. They can do by themselves at home or anywhere. High sensitivity means that the sensors must be sensitive enough to detect, especially with a low amount, very little amount of sample. For example, if we want to detect certain disease, if the sensors have the high sensitivity, high specificity, we can detect at an early stage, for example. And they have a potential ability for real time, so can check it straight away, and can give the reading, can give the measurement straight away, and on-site analysis, fast and low cost. Of course, this is all the factors that for me is quite ideal. It's quite difficult to provide or to develop sensors that have all these advantages. So biosensors, actually, there are a lot of applications. When we talk about bio, is everything in our life. We are reacting with biology. For example, we eat food. Food is all about chemicals, all about biology. So we need to test, we need to analyze the food, we need to have a sensors in the ingredient, for example, either the food was contaminated or not. So this is all need sensors. Environment control, to measure our quality of our air. So we need a sensors. Clinical detection for a disease, as I said. For drug and agriculture industries, and there are a lot more actually. We require biosensors, the sensors that exploited biological or biochemicals to give the signal. New generation of biosensors combining new bioreceptors. Nowadays, biosensors, when we talk about bios, sometimes, people are clever now. They can develop synthetic biological or biochemicals. It's not only the pure biochemicals, but also the synthetic one also available, which actually react the same as the pure biochemical and give the reaction exactly the same. For example, now we are talking about antigen antibody, but actually people are developing aptamers, for example, to replace antigen antibody. Although it's not quite similar, but it gives a similar response. So that's about bioreceptors. And there are also people working on the signal amplification. When we talk about signal amplification, that it has two methods actually. Either we are using new materials, new substrate to have more interaction with the chemicals, or we are using particles to amplify the signal. So this is really depend on what we are going to detect, and is it either suitable or not. There are a lot of several factors that we have to look into in order to produce a good biosensors, and with the very ideal advantages. Sometimes the sensors no need very sensitive. For example, for the disease, might be people don't want to have very sensitive, because if there is very sensitive, it make them feel not well, or not good, because it's very sensitive. It might be wrong signal as well. So it doesn't, for the sensors in to detect a disease, the difference of 0.1 or 0.01 is really didn't make sense for the user to compare with. So oops. So despite the fact that there are high numbers of papers published very few are commercialized in market. This you know, is normal because yeah, they are not easy to commercialize, especially when we talk about biosensors. But there are a lot of research working on that. There are people working on different methods. There are people working on different platforms. There are people, researchers working on modifying the surface. There are a lot of papers published, and the paper for biosensors, you know, is very high impact factor. For example, if you publish in biosensors, bioelectronics, for example, I think now the impact factor is about 10, if I'm not mistaken. So in the past, 7 or 8. So that's what it's all about, because when we talk about biosensors, if we want to commercialize, it require clinical testing that might, you know, give the limitation of the biosensors that want to go to the market. Okay, and then biosensor, they have analyte, they have bioreceptor. For example, they have a pair. There must be a pair. For enzyme, they have a substrate as the analyte. There are a lot of sensors that use enzyme. For example, the very easy one is to check glucose level. They use glucose oxidase. So when there is an ACE at the end, it is an enzyme. So glucose oxidase. Okay, and then antigen and antibody. Antigen is the analyte, antibody is the receptor. This actually, you know, same as our body. When the foreign things go into our body will produce antibody to capture the antigen. So this is very specific. Antibody for antigen, for example, if the antibody for dengue fever, they will catch on the antigen for dengue fever. And then DNA, single strand. So our DNA, they have 2 strands. Our DNA have 2 strands. So these 2 strands we can have hybridize, give the higher temperature, so it becomes single strand. And this single strand can only bind with the same strand, their pair. So they will not bind with another pair of DNA. And as well as protein-protein interaction, there are a lot of protein-cell interaction, but it's very specific. The important thing, analyte and bioreceptor must be very specific binding. So in this figure, if you look at the bottom one, so we have the analyte, we have the bioreceptors. When the bioreceptors react with analyte, so it will transduce, and they will give a signal to the transducer, and they give a readout. So another figure shows that there are several types of, as I mentioned just now, in Y shape here is the antibody. So enzyme is like a protein here. And we have single strand DNA, where if we want to use all these biological, all the biological things, we need to really immobilize or properly deposit all these biological component on the solid surface. That is the challenges that the researchers have to face, how to make sure that the antibody are in the upward position, the enzyme is still working, the DNA are properly deposited on the solid surface. Okay, I show some of the various type of antibodies. This, when we use antigen and antibody as in our sensors, in our biosensors, and then we call it immunosensors. It's immune sensors, immunosensors, which consists of the interaction of antigen and antibody. So there are 5 types of antibody in our body. We have IgG, which is normally we heard that IgG, IgD, IgE, IgA, and IgM. Yeah, and IgM. So the Y shape is represent for antibodies, and the FC is a bottom part here, is the lower part, and the FAB, 2 shape here, Y shape, is the one that will capture the antigen. If the one that will capture the antigen. So if we look at, if we compare IgG and IgM, so IgM have more binding site as compared to IgG, but IgG is the most abundant in the serum, our serum immunoglobulin. So in our blood, in our body, they are abundant of about 80% of IgG as compared to IgM or other antibodies. So this is quite important, basically, because if you want to use or exploit antibody for the interaction, we have to look at the possible or the right type of antibody to be deposited on the surface of biosensors. Okay, this is as I said, this is challenging because if you deposit antibodies on the solid surface, there are several positions that might happen. First is the end on here, and then the second one is the side on, may lay down, and then head on. So the head is facing towards the solid surface, and then lying on, they will lie on the surface. So this actually, if we didn't do it carefully or didn't treat it carefully, so the reading from our sensors might be give a wrong signal. So the binding may not happen on the specific antibody, but that might bind or they go to somewhere else. So the best one is the end on position, where the binding site facing upwards. So where here we will inject or introduce antigen and will bind at the binding site of the antibodies. So how to do that? How to make sure that the antibodies is deposited at the upward position and on position. So to do that, normally I use protein. So other protein, protein A or protein G, to help the antibodies deposited at the end-on position. So in this position, it will optimize the interaction of the antibody towards the antigen. But other factors that have to be considered when we deposited or immobilized the antibodies on the surface is about the pH. We have to look at the right pH, the right isoelectric point for the protein in order to ensure that the charge of the protein is at the right one. And then we have to careful about the temperature, because antibody is a part of a protein where it can change the conformation at different temperatures. So we have to ensure that we are working at the right temperature, means that the temperature must not exceed 25 degrees Celsius in order to to maintain the functionality of the antibody. And then the functional group as well. When we want to immobilize the antibody on the surface, we have to look at the functional group of the whatever biochemicals that we use in order to ensure that all these chemicals or biological element that we use are properly and are in a working condition. Because all this we can't see in bare eyes. And we just look at the signals. Either if we immobilize it properly, then only we can get a good signal. If not, then we are not sure what will happen, what actually happened during the immobilization process. And then there are three types of binding assay. Three types of binding assay, either direct or sandwich or competitive. Direct is very easy, where we immobilize the antibody or we immobilize the antigen, and then we introduce either antigen or antibody for the same disease or same type of antigen or antibody. So it will react, and we will get the higher concentration will give the signal, higher signal, and then after sometimes it will saturate because of the binding site is already full. For the second one is a sandwich assay, where we introduce another antibody. We introduce another antibody to bind and to amplify the signal, 2 or 3. But if we want to use more than one antibodies, we have to make sure that we use different antibodies. For example, if for the first one is anti-rabbit, the second one must be anti-mouse. So if not, then it won't bind. So that is basically what we need to know first. And then for the sandwich, actually they have more better signal because of the, there's amplification here using more antibodies. So if we compare with direct, sandwich assay is better in terms of the signal. And then we have the competitive assay. So the competitive is actually, we have inhibited antigen, where this antibody will compete with the antibody that we immobilize on the surface, and the higher concentration of antigen will give the lower signal. So if we compare competitive and sandwich, it gave a high signal, but it reverse. For the competitive, the lower analyte concentration will give higher signal, but for sandwich, the lower concentration of analyte will give a lower signal. So this is basically the binding assay that we need to consider before we develop the biosensors. And the measurement protocol for antibody is, when we want to deposit the biosensor, we want to work with the biosensor, there are 2 stages. One is we have to immobilize the antibody first. So we call it antibody immobilization stage, where the antibody is deposited through layer by layer deposition of biochemicals. We cannot straight away deposit the biochemicals on the solid surface. We need to treat the solid surface through using the biochemical first, and then only we immobilize the protein G or protein A, and then on the antibodies. So there are steps that we have to follow in order to immobilize antibody on the solid surface. So the measurements are taken after each deposition in order for us to ensure that all these biochemicals are properly deposited on the surface. And then for immunoassay, that is actually after we immobilize with the antibody. So now we want to test either our sensors react with the right analyte. So we have to do the dilution of the analyte concentration Let's start from low concentration to the higher concentration, and then we inject to our substrate, which consists of antibodies, and we do the measurement after each of the concentration. Sometimes we can do the surface regeneration, or we can without surface regeneration. I will explain later. So method of detection, either optical or electrochemical, but electrochemical is more popular. So optical is normally for the lab-based method, but recently there are also optical fibers method where we can produce it, the sensors as mobile sensors. But for optical, for example, SPR is surface plasmon resonance. So this is the picture, surface plasmon resonance. Here is about looking at the right angle in order for the light interact with antigen and antibody or the biochemical on top of the surface. So it will give the signal in terms of the ratio of intensity. So number two for optical method of detection is about ellipsometer. So ellipsometer, they are using elliptical polarization of light source, and then they collect the reflection of the light. So this one is more sensitive. It gives the output in terms of phase shift and as well as the ratio of the intensity. There are two types of output for ellipsometer. And then optical fibers bottom. So if you want to work with optical fibers, the challenges is about to open the cladding, to slice the cladding, and then to deposit several biochemicals on top of the core. So this is very tiny, and we need to do it very carefully because the concentration, and as well as we need to have liquid cell here in order for us to inject all these biochemical on top of the core of the fiber. And then for electrochemical, this is actually the potential state where it's all about the electrolysis process. We have the 3 different electrodes, and now we make it smaller, like a screen printed electrode, and then we measure cyclic voltammetry, amperometry, and a Nyquist plot, for example. So that is electrochemical. If we compare electrochemical, optical method is quite higher, more sensitive, and as compared to electrochemical, but electrochemical is more easier to handle. And the advantage of optical is there's non-contact. They didn't straightaway contact to the liquid that we are measuring. Okay, this is what I say, that if you want to amplify the signal, first is about the surface modification. So when we do surface modification, actually we want to have more binding site, for, more binding space for the antibody. As for in this picture, this is a porous surface, so the antibodies can easily bind. The FC of the antibody can easily bind. There are more spaces as compared to the flat surface. So we can use new substrate or new materials for the electron in order to give more binding space for the antibodies. And second thing, by exploiting nanoparticles, metal nanoparticles, there are a lot of work working on this. when we have nanoparticles, means that we have more area, more surface area for the biochemicals to interact. Yeah? From the bulk materials, so we are using nanoparticles, so there are more surface area for the biochemicals to interact. And then by conjugation, so conjugations means that we attach the antibody on the nanoparticles. So instead of we have one antibodies, if we use nanoparticle, we can have many antibodies attached to one nanoparticles. So instead of one surface, but when we have introduced nanoparticles, we have more surface area for the biochemicals, for the antibody to bind. Second is about the ellipsometry. As I said, there are two parameters that can measure using ellipsometry. So first is psi, which is the ratio of light intensity, and then the second one is the delta, which is the phase shift of the light, of the reflected light. So for ellipsometry, they measure the optical properties, especially the thickness, refractive index, and then this is actually ellipsometry is actually for the purpose of measuring the thickness in the nanometers thickness, but normally familiar in semiconductor industry. But in order for us to use in biosensing applications, we need to do some modification. So I did in the past, modify the ellipsometry in T-ray configuration, total internal reflection ellipsometry, in order to detect the different thickness after each of the interaction of biochemicals. They can measure two measurements, static measurement, psi and delta, and as well as kinetic measurement to measure association and the association constant of the interaction. But the disadvantage of ellipsometry is, it require fitting. The experienced people to fit the data to get the thickness, because the thickness is very, very small. And then now electrochemicals, there is a potential stat here. So this is the blue bottom here. This is the conventional where previously we used as a electrolysis process, but nowadays we can use a screen printed electrode here and then deposit all the biochemicals on this small area, and then connect to the auto lab, connect to the equipment to get the signal. So it's very easy, and then it's very mobile. So it measures the electrical properties based on the chemical interactions, for example, impedance, ampere, volt, after the biochemical interaction. Suitable for point of care, where we can use it everywhere. No need experienced people. After we do all this calibration, we can connect to the reader. Not very accurate as compared to optical method, but it's comparable. It can be used for ordinary people, because when we use electrochemical, electrical method, there must be a noise of the signal, and then if we compare with optical, of course, they're slightly, in terms of the sensitivity, electrochemical is less sensitive as compared to optical method. This is the example of my work by using ellipsometer. So in this work, actually, I detect aflatoxin, mycotoxin is very small molecules, where sometimes for electrochemical method cannot detect, or the other methods is very difficult to detect. So in this case, if you look at the graph here, so I need to deposit another polymer, pH. So the first layer is the gold layer, and then the second one is the polymer to treat the surface. The third layer is protein, as I said, to help the binding of antibody. And then number 4 is the antibody for aflatoxin, and number 5 to number 9 is different concentration of aflatoxin. So this method able to detect down to 0.04 nanogram per mil of aflatoxin. And the second graph here is typical of the different thickness after different concentration. So we can see that this is the logarithmal scale concentration. At the higher concentration, the thickness is quite saturated because of the binding site is already full. So that's why there is no difference in terms of the thickness. That one. So this is actually the static measurement that we measured using ellipsometer. But ellipsometer can measure the kinetic as well. So for the whole range of spectroscopic wavelength, so we can measure the kinetic during the binding event. So this is the example of one of the concentration of aflatoxin. And then from this data, actually we can manipulate. We can manipulate the S, actually the time constant versus concentration. So we get the equation where from here, from this equation, we can calculate the value of association and dissociation constant. So if we look at here, 2.57 power of 6 is the association constant, where it's typical for a monoclonal antibody interaction. So antibody, they have two types. One is a polyclonal, another one is the pure antibody. We call it monoclonal antibody. And then this is another work. This is another work where I need to increase the area of binding site for antibody. So I try to introduce new material, polyaniline dope iron oxide nanocomposite for the detection of amyloid beta 42. This amyloid beta 42 is actually the biomarkers for Alzheimer's disease. So there are all the chemical process here to produce nanocomposite. As you can see here, they are very small materials, composite that we managed to produce on the surface. And then as well as the antibody number 4. Number first is the bare substrate FTO. Number 2 is the PANI Fe3O4 nanocomposite. And then D is after we immobilize the antibody of the amyloid beta. So this is the signal of the impedance gathered from the electrochemical method. And then from the electrochemical method, we can measure the cyclic voltammogram of anti-amyloid beta immobilization stage. So this is the immobilization stage for the different biochemicals. And then the bottom one is actually immunoassay of the amyloid beta 42 from nanogram per mil. You can see the difference, and here the red color is about it's a typical result where after at a higher concentration the signal will saturate. And then another work is going on about the prostate cancer. This previously I work on this one. So all the previous work is actually using direct immunoassay, where I immobilize antibody and then I inject different concentration of antigen analyte. But this one is a slightly different. It's a competitive assay, as I explained earlier. But I'm using a quartz crystal method, quartz crystal microbalance method for this type of biosensors. It's a very nice result where we can see, normally at a lower concentration, if I'm using direct, at a lower concentration, the signal is low, and it becomes saturated at a higher concentration. But in competitive assay, at a lower concentration, the impedance the signal is higher and become saturated at a higher concentration. So in this work, actually quartz crystal microbalance method, we are measuring the impedance after the binding of all these biochemicals. As you can see here, so we have the reference air, measure the signal in air, measure the signal after each of the deposition antibody. BSA is bovine serum albumin, in order to cover the surface after we immobilize the antibody. And then the second one, the bottom part, is about the different concentration of antigen introduced on the surface. Okay, this is all about my work, all about, I already talked about the fundamentals of biosensors. I talk about the method of biosensors. I talk about my work, my recent work. And actually, I have also the previous work about the heat shock protein and so on. So, and then now it's about COVID-19, where I think is a lot of people work on it. It's a very interesting topic, and there are a lot of funding on this work, this research, because it's very demanding now. A lot of people want to produce it, very fast, very soon. Very required throughout the world. So it's quite interesting where the Harvard and MIT researchers, they are, instead of using other materials, they are using the face mask that we wear, where it can produce a signal when there is a virus. That is very interesting. It might use antibody as well, but how it embedded the antibodies on the face mask is also questioning. But this work actually is not finished yet. They are working on it because they worked for the other virus in the past. But it's very interesting where if there is a virus, attached on the mask, or when the people breathe or sneeze, it has a signal in terms of the color, different color. Okay, there are also a paper-based detection. This is also a work by MIT researcher, where there's a special paper. You can see here they mobilize the antibodies on it, and then when the antibodies react with the virus, they will bind and they will give the signal. So This is actually very interesting to look into how the antibody can be immobilized on the solid surface. You know, as we know, it's not easy and quite challenging, and you know, exposed to the ambient. Yeah, what I want to show here is about, you know, previously I explained about the various type of antibodies. Now you can see people talk about IgM and IgG. So IgM and IgG. So why IgM has give a very good response. You can see here, if your onset symptoms after 3 days, you know, IgM senses more, give more response, more faster response as compared to IgG. It is because of IgG has more binding site as compared to IgM. IgM has more binding site as compared to IgG. So IgM has five antibodies, where IgG is only one. So that's the reason sometimes people talk about it. So, well, it's not, when we talk about the selection, the right selection of the chemicals of the antibody, so we have to look at the necessity and the cost of the production to use it. Sometimes it's very good response, but the cost is very high. Also, it's not suitable to be commercialized, because IgM is very little in our body, but because of the abundance of IgG instead of IgM and other antibodies type. There are a lot of antigen antibody test kit for COVID-19. You can just serve. This is not, it's not mine. So all is about using antigen antibody test, antigen antibody interaction. Challenges forward is to improvement of sensitivity and specificity. All the researchers, you know, working on the high sensitivity, because of you know, in order for the disease to detect at the early stage, it need to very sensitive, but must be suitable, must be very specific to the disease. That's what's all about. And to miniaturize of sensing platform and multi-spot. And now people like to use very small, you know, platform, because it's easy for them to measure anywhere, anytime, at home, for example. And the best biosensor, for me, if the biosensor, the sensors can detect multi disease, for example, for one spot, they can detect sugar level, they can detect uric acid, or they can detect whatever, you know, disease at the early stage. And then point of care biosensors, again, we need to have a sensors where people can easily use it anywhere without having any experienced staff or experienced person to diagnose, to measure, to do the measurement, and must be disposable and low cost. So looking forward for biosensors, there are actually this area actually required a lot of researchers collaborations, because biosensors is actually combined of multidiscipline, various disciplines of the researchers. And you know, in terms of disease, when we talk about disease, there are a lot of disease all over the world. Even now, there are new disease coming out. So there's no end for biosensors area. So with that, I would like to end my presentation. I open to the floor for if there is any question. Thank you very much.
Thank you, Professor.
Yeah.
Thank you for your valuable time to giving this presentation. It is very nice presentation to the session. Now I request Professor
Sir,
Dr.
one second, sir.
Yes,
A question, sir. There is a question, few questions.
Yeah.
So before that, I want to thank you, sir. A very beautiful session,
Yeah.
a wonderful session,
Okay.
and you have beautifully explained so many things, which is very relevant to current scenario. This is a wonderful session, sir. So if anyone have any questions or doubts, you can ask our resource person now.
Say again.
If anyone have any questions,
Any?
okay, sir. Let me ask Dr. Selvi. Want to ask a couple of questions, sir.
Yeah.
Whether disposable biosensors are easily degradable?
Ah, that's so far no. I can, to be honest, it's not biodegradable. Maybe we can, you know, for the next challenge is to produce biodegradable biosensors. So far, to be honest, the sensors is normally either made from polymers, from carbon, yeah, from actually the plastic base. So I didn't see so far that the sensors might be later on using paper. Yeah, that's coming up. People using paper, as I showed just now. MIT researchers working on paper-based biosensor. That's very interesting to look at it. When I think papers is biodegradable as compared to polymers that we use now.
Thank you, sir. Another question. What is the range of thermal or heat liberated while using biosensors like burglar's alarm?
Say again. What is the?
What is the range of thermal or heat liberated while using biosensors like burglar's alarm? Alarm.
Okay, okay. That one is different kind of sensors, because this is about the heat sensors, or it's about the infrared sensors. So infrared sensors actually they are slightly different on the biosensors in terms of the working principles. Well, for the burglar's alarm, normally if you are using infrared, it will be enough, because it doesn't require a very sensitive. It just need a movement, or they just need a body temperature. Body temperature of the human is quite standard. So yeah, I think that one might be a different kind of method of detection for burglar's alarm.
Thank you, sir. Another question from one of the participants, sir, asking about how can we reduce or neutralize the sandwich property of antibody to get balanced signals?
Okay, the sandwich assay, eh?
Yes, sir.
Okay.
Asking about how can we reduce or neutralize the sandwich property of antibody to get balanced signals.
To get a balance?
You want me to repeat it, sir?
Yeah,
balance signals.
Balance signal. Okay.
Yeah.
Well, first of all, if we use antibody, at the moment the antibody is quite expensive. For example, the one that I use, 1 milligrams around 1,500 ringgit. So in order for us to get, you know, to use antibody, or to exploit the antibody, for our biosensors, you know, we need to do it very carefully. We need to have more experienced people, you know, in the first place, in order to use antibody for biosensors. especially to select which type of binding assay that we want to use. Either we want to use direct, either want to use signal sandwich, or we want to use competitive. Because if you want to have the good signal, the high response, you need to have more antibodies. But if you use more antibodies, the cost will be higher. So you have to balance in terms of what is the application, and then the cost that you need to cover. Yeah? But sometimes people don't care about the cost because of, for example, during COVID-19, the pandemic is all over the world, where it costs more, a lot more as compared to the price of the antibodies. So people don't care either using sandwich or using competitive assay, as long as they can detect in an early stage. And then the detection sensitivity is very, very important as compared to the cost. So that's why when we want to develop biosensors, there are several factors that we need to consider. It's not only the cost, also the significant or the necessity of the sensors during the time.
Thank you, sir.
Yeah.
Another one question. Is there any research have done? 1 second, sir.
Research have done on?
1 second, sir. 1 second. Is there any research have done to diagnose COVID-19 using biosensor?
Yes, people working on it. Actually, there are in other group. I'm a member of a Sensor Malaysia group research on biosensors. In Malaysia, there is a group working on it, COVID-19 biosensors, and I think in US, in all over the world, actually working on the detection of COVID-19. So the one that I show is a very good example where people, researchers from MIT and Harvard, actually they are developing the wearable sensors embedded at the face mask. The face mask actually is disposable, where people wear it and then just throw it away. But when we can enhance the face mask to detect the virus, is very, very good. There are two things that we have to think about this virus. First is to detect, for the detection, that require biosensors, the detection. And then second one, to prevent the disease, where people look at the vaccine now. So I think India working on co-vaccine, they call it co-vaccine. US, they are working on Moderna vaccine for COVID-19. So first is detect, the second one is to prevent by using vaccine. So my answer is yes. A lot of people, a lot of researchers now, if you want to explore the new area of research, or you want to produce a good paper, that's the time you have to explore on COVID-19 research, because of this is new things that we can explore.
Thank you, sir. One last question, sir.
Yeah.
Is there any portable biosensors available to check prostate cancer, sir?
No.
So far there is in the market where the prostate, you can use the urine. You can use urine to detect the prostate cancer, but it's still not good enough because of the sensitivity. It's not good, and normally it's misinterpretation because of the specificity of the sensors. People work on it to improve the sensors to have highest sensitivity, because normally for prostate sensor was detected at the severe stage, level stage 3, stage 4. So there is a biosensors for prostate, but it's not good enough at the moment.
Thank you, sir. I hope,
Madam,
your
Madam,
doubt got clear.
Madam.
Yes, sir.
Yes, sir. Yes, sir. Thank you very much, Doctor, for the nice presentation on biosensors. I am from Department of Pharmacy. I would like to ask about a very nice introduction class for me, sir, I could say, because I never exposed to biosensors before. Thank you very much for that. And I would like to know, are there any expiry date for this? Because in market we have seen uric acid, sugar level and hemoglobin biosensors are available. Do they have any expiry date? In India, we all use it until it gets faulty function or it break. Otherwise, we don't dispose it. So I would like to know about the expiry dates of these gadgets, these biosensors.
Okay. Normally, for that kind of sensors, you know, they are using enzyme. Enzyme is very stable. Enzyme is very stable, but it cannot reuse. Once it interact, when it's already exposed to the substrate, when it's already used, they cannot reuse it again. So this is like a chemical reaction. Once it's they interact, and then they, you know, they produce when the enzyme react with substrate, they will produce the new product. So you cannot reuse it again. So so there is no expiry date, but once you use it, you cannot reuse it again. So that's the problem of you know, current biosensors.
Thank you, sir. Thanks, sir.
Madam, madam.
Sir?
Yes, madam. Anybody asking questions?
No, sir. That's all. We came to the today's session. Today, both the sessions were very informative and went well. Again, we shall meet tomorrow morning, third session at 10:45 a.m. Thank you so much, sir.
Okay, thank you very much. Thank you for the organizer.
Thank you
for all the questions raised from the participants. Hopefully, the conference will go very smooth and very fruitful conference. Thank you.
Thank you, sir. Thank you so much for being with us, sir.
Thank you,
sir. Thank you, sir. So, dear professors, kindly give your attendance, please, to chart your name and affiliation. Please, we are waiting for another 5 minutes to kindly put your attendance, name and affiliation.
Hello, sir.
Yes, sir. Yes, professor.
Sanatan Singh from Johor University, Odisha.
Yes, professor. How are you? Professor.
Fine, sir.
Yes, professor.
Hello, sir.
Yes, professor.
Yes, sir.
Hi.
That session is very interesting, sir. Hello, sir.
Yes, sir. Please. Please, sir.
Kamalakandan, sir.
Vanakkam, madam.
Is there any feedback link? Naanga chat box la pair alla potutom.
Yes, madam. Madam, feedback on the final day da, madam. Namba over session paakradillinga.
Seringa.
Final day kudupom, madam.
Naleka.
Ama, madam.
Okay. Thank you, sir.
Adalla mention pannila, madam.
Sure, sir.
Right, madam. In between நீங்க உங்களுடைய சஜஷன் அப்பப்ப சொல்லலாம், madam.
கண்டிப்பா சார், நல்லா இருந்தது. அது எஸ்பெஷலி இது பயோசென்சர்ஸ் ரொம்ப நல்லா இருந்தது சார். அது ஒரு யூஸ்ஃபுல்லான ஒரு இது இருந்தது. குட் இனிஷியேட்டிவ் சார்.
தேங்க்யூ மேடம். தேங்க்யூ மேடம். Sanandan Singh sir. Sanandan Singh. மேடம் வெற்றிச்செல்வி மேடம்.
ஹலோ சார்.
மேடம் க்ளோஸ் பண்ணிடலாங்களா?
க்ளோஸ் பண்ணிடலாங்க சார். க்ளோஸ் பண்ணிடலாம்.
ரைட் மேடம். உங்களோட சஜஷன்
முடியலைங்க சார்.
ஏதாவது குறை நிறைகள்?
இன்னைக்கு செஷன் நல்லபடியா முடிஞ்சது.
குறை நிறைகள் சொன்னீங்கன்னா நான் நெக்ஸ்ட் வீக் ரெடி பண்றதுக்கு.
பார்ட்டிசிபென்ட்ஸ் எல்லாம் அவங்க சஜஷன்ஸ் ஃபீட்பேக் கொடுப்பாங்க இல்லைங்க சார். என்ன அவங்க எல்லாம் இருக்காங்களே?
இருக்காங்க மேடம். இப்போ அட்டெண்டன்ஸ் போட சொல்லிருக்கீங்களே, கொஞ்சம் அதை முன்னாடியே சொல்லிடலாம் மேடம். ஏன்னா பாதி பேர் வந்து போயிடுறாங்க.
சார்
சொல்லுங்க மேம்.
சாரி, இந்த கரண்ட் போயிடுச்சு, அதனால லேப்டாப்ல கனெக்ட் பண்ண முடியல. சாரோட மொபைல்ல இருந்து பண்றேன். ஒரே சின்ன இது சார், உங்களோட அந்த பிரசன்டேஷன்ல வந்து ஒரு ரெட் கலர் சர்க்கிள், ஒரு லைன் மாதிரி ஒண்ணு வருது சார் எல்லா ஸ்லைட்ஸ்லயும்.
ஆமா, எல்லாரோட ஸ்லைட்லயுமே.
மேடம் அது ரெக்கார்டிங் மேடம். அது
ரெக்கார்டிங் இல்ல சார், அது ரெக்கார்டிங் பட்டன். நான் சொல்றது அந்த நடுவுல ஒரு பிரசன்டேஷன்ல ஒரு இது மாதிரி வருது.
இல்ல அவர்
சும்மா லைன் போட்டு மார்க் பண்ண மாதிரி ஒரு இது வருது.
வருது.
அவர் கொடுத்துருக்கா மேடம். அது இண்டிகேஷன் மேடம்.
ஓ, அவரோட ஸ்லைட்லங்கறீங்களா?
ஆமா மேடம், அது இண்டிகேஷன்.
அது எல்லா ஸ்லைட்லயுமே இருந்தது, எனக்கு அதான் ஒரு டவுட்.
இல்லங்க, அவர் இண்டிகேஷன் கொடுத்துருக்கீங்க அது.
சரி சரி.
இல்ல சார், ஆக்சுவலா அந்த ஸ்கிரிப்லிங் வந்து வியூ ஆப்ஷன்ஸ்ல வந்து அன்னோட்டேஷன் ஒன்னு இருக்குங்க சார். வியூ ஆப்ஷன் நமக்கு மேல ஸ்கிரீனோட மேல பாத்தீங்கன்னா வியூ ஆப்ஷன்ஸ் இருக்கும். அதுல போய் அன்னோட்டேட் அப்படின்னு போட்டு அதுல வந்து ஸ்கிரிபிள் பண்ணுவாங்க. யூசுவலா ஸ்டூடெண்ட்ஸ் வந்து ஸ்கிரிபிள் பண்ணிடுவாங்க. சார் ஆக்சுவலா ஏரோ மார்க் கொண்டு போகணும் அப்படின்றதுனால அவருக்கு வந்து ஸ்கிரிபிள் ஆயிருச்சு. அது ஆக்சுவலா அண்டர்லைன் பண்ற மாதிரி ஸ்கிரிபிள் ஆயிடுச்சுங்க. அது வந்து அங்கேயே கிளியர் இல்லைன்னா அண்டுன்னு ஒரு ஆப்ஷன் இருக்குங்க சார். அதை பண்ணா அது கிளியர் ஆயிரும். இது ஹோஸ்டோ இல்ல கோ ஹோஸ்டோ மட்டும் தான் பண்ண முடியுங்க. மத்தவங்க எல்லாம் கிறுக்கலாம், they can scribble, but maybe some they cannot clear it, but the host can clear it, sir.
ரைட் மேடம்.